So thank you everyone for attending today's talk regarding nanopore sequencing and different library chemistries we have which you can apply. Um, so today, at least at the first hour today, I'm going to give you an overview of how nanopore chemistry works, a little bit about adaptive sampling, different type of libraries you can do using Nanopore, and then specifically in the cfDNA context, what are the QC measurements which are required before you move to the library preparation phase. And then we'll talk about base calling model accuracy. And finally, and I think that part I'm going to do after, we review yesterday's run, discuss the run reports. In the afternoon, it would be more about our software, and then the downstream analysis, which we can do using Nanopore, and that software is called Epitome. I think tomorrow, we'll see based on how the run goes. If the run is done and finished, we can try to install the Epitome. I have already shared the protocol for Windows system, how we can install that container. And that Epitome container has actually different workflows. So we can at least install the relevant workflows which you can use for the cfDNA sequencing analysis. And tomorrow, I have also invited our bioinformatics scientist, and we are going to talk more about the cfDNA specific analysis, including methylation calling. If we do methylation calling, what would be the output? Because the output format, like the BET file format, would be a little bit different. And we'll also try to do at least a demo data if we cannot run the real data tomorrow, how we can, download that data and how we can do the epitome workflow and, get the data result out of it. And I think we should also probably meet in a couple of weeks and do, the data review from yesterday's run, or if you can do another run with a methylation calling on. So everything we should do data review. Another suggestion would be, and it depends on your lab. Usually, what we do, like we try to meet monthly, just to get an update of all the experiments, what you are doing, if you need any additional help, and also specifically for the data analysis part. It could be very short, but it is always very helpful to have a monthly meeting. So that's a suggestion we have from our side, and you can think about it and let me know if you think it is a good idea. Okay, so let's start. I would again start with how nanopore sequencing works. And as you already know now, at the heart of our sequencing, it is the nanopore, which is a nanoscale whole protein complex, which we see naturally in like different biological systems, example across membrane. And in ONT space, we have actually utilized those naturally occurring nanopore, enhanced its functionality by introducing some mutation in the barrel region of the nanopore and inserted it across lipid membrane and made it a system which is kind of a molecular culture counter, and that is what our sequencing platform looks like. So we talked about it yesterday, so I'd be very brief today. So how this system really works. So this blue part is the nanopore, which is embedded in this gray lipid membrane. DNA or RNA it passes through the pore due to a difference in electrical energy or electrical potential. And the movement is controlled by this motor enzyme. And for Nanopore, it does not matter what type of library you are preparing, the addition of the motor enzyme is basically the library prep. This motor enzyme is different for DNA or RNA, including the interflow cell that is different for RNA and DNA. And the enzyme controls the speed of movement of the native DNA or RNA strand. For DNA, it is close to 450 bases per second. For RNA, it is 120 bases per second. So what happens when a native strand passes through the pore? The electrical energy, there is a disruption of the electrical energy, which we call squiggle, you can see here. And in using our software, which is called Nano, which is trained using like different models, like it's a machine learning or neural network based software, we can deconvolute, this signal and get the information of all the bases at a single nucleotide resolution.
Does a specific base have a specific signal?
Yeah. So this is how the base calling algorithm works. So the main features of our sequencing platform is, as you can see, it is a direct sequencing of DNA, RNA. It is a PCR-free platform, so there is no amplification biases, and because of this, the base modification signals are contained. So here also want to mention, because I think you mentioned yesterday, what if we have really low input samples? So we do also have protocol for low input samples, but that time you have to include PCR. So there would be, all the whatever we know the biases coming out of PCR, and also we are not going to get them this modification information. So it's a read length agnostic platform. Nanopore started as a long read sequencing platform, but presently it is a read length agnostic. So for DNA, I think we can go as low as 50 bases, as large as few megabases in length. It's a real-time analysis because of the Mino software, which is doing all the the deconvolution at this in the real time. It is very scalable, and it also presents as a low-cost format, specifically when we are using platforms like Nina.
Sorry, I have a question at this point.
Mm-hmm.
So how is it different, the motor enzyme works between DNA and RNA? So DNA is double-stranded, so here it works like a helicase concept, right?
Yeah.
RNA is single-stranded. So how do I go through the motor enzyme?
The helicase activity is so the motor enzyme is different. It's more for a the motor enzyme is also attached to an adapter molecule.
Mm-hmm.
I think yesterday I was showing a diagram, which is a single nucleotide region.
Okay.
So it is kind of acting as also a tethering platform. So it is helping the DNA or RNA to come in close proximity with the nanopore. And for RNA, it is controlling the speed.
So there is no seed DNA library there. Even if the starting material is RNA, there's no CD.
So you can do two ways. If you want to do direct RNA, which means you don't need to have cDNA. You are, sequencing the RNA directly. But there is also possibility that you can do reverse transcription and then you read the cDNA.
So
for that, it is DNA platform. Because it is no more RNA.
Yeah.
But you are getting, it's the transcriptomic
the expression.
You get the transcriptomic expression like in other platforms you get it. I will also briefly show you what are the different RNA sequencing specific library prep we have. In future, if you're interested to explore, yeah, you can. So I want to briefly mention about this chemistry. It is called adaptive sampling. And this is quite interesting, and this is also working quite nicely. So this is, I think, more important in the context of targeted sequencing. So example, okay, first let me tell you what it is. So adaptive sampling is basically a software-based enrichment of a specific region of interest. So what we are doing, so it is the same, chemistry we are utilizing here. So the nanopore chemistry did not change. Library prep did not change here. What we are giving additional information to the software is a bed file. The bed file has all the information of the region of interest, which we want to amplify. So in the software, what it can do, it can reject all the strands which are not part of the bed file, and it can only sequence the specific region of interest. In that way, you are getting an enriched information of your target. It might not be an important application for cfDNA, but when we are thinking about gDNA, like the entire genome of interest, and we are specifically interested into, let's say, a particular group of genes. Specifically, I have seen like in oncology labs, oncology clinical labs, they're using this application a lot because there is no need of now doing extra bench work or wet lab part for enriching a particular targeted region. You can just
software based.
This is a software based, but you are just supplying a big file to the software, and then the software is enriching, all the genes you are interested in, and this is working, extremely good. So, and also very user friendly to use.
Yeah.
And in the downstream analysis, we have seen like great accuracy for all the variant calling, even with adaptive sampling. So, what it means, like the entire platform is working quite perfectly. So if you ever in future interested in, employing this, in utilizing this adaptive sampling, let us know. We can help you with to set up this adaptive sampling. So it is also like in our Mino software, there is a toggle button. You can just click that. It says adaptive sampling on, and then it will ask you like provide the FASTQ file, provide the bed file information, and that would be the extra additional information we need to provide. The library thread is going to be the same, which is LSK 114. Okay, so a little bit about, different devices we have. Example, in your lab, you have P2 Solo. This is the platform you have, and the flow cell for the P2 Solo is PromethION, which we saw yesterday. So PromethION can be used in P2 Solo, P2I, and P24. So these are the three platforms we have. This is like the highest output platform. As the name suggests, there are like 24 slots. P2I, it has 2 slots, but the computational additional space for P2 Solo, we had to figure out like, which would be the perfect computational part. For P2I, we don't need to do that. It comes within. There is another platform in terms of flow cell. It is called MinION. So MinION can be used in MK1D, and it is like, it just looks like this cell phone, and I thought of bringing it today, but I forgot it at home. So you can just take it to any, it is very much useful, I think, for all the groups who are doing field work in many secluded regions, even we do have projects with NASA, like they do it in when they're going out of space. So this is where they do the sequencing because it's so user friendly. And this is GridION, and this is like more like a robotic system called ELISION. So in the GridION, also the flow cytotropin ion, it is very much, I think, useful in microbiology space. And these are like the lower throughput like flow cell compared to PromethION. Okay, so
how long does it take to sequence in?
it depends like what type of sequencing you can do. You can do like longer sequencing, but the flows, there are not that many pores. So I think usually a day, that's what maximum people are running. So the P2 solar, I mean,
can I join in from the outside here? I've missed a lot of this, but I'm wondering, we have some extracellular vesicles, enriched extracellular vesicles isolated that we want to do RNA sequencing on. Could we use this this platform for that?
Which platform? You mean the P2 Solo?
Well, whichever, yeah, any of them. Yes, the one we have.
Yeah, so here you have P2 Solo, and that is a perfect platform for doing any type of RNA sequencing. Now, I will show a little bit about, I'll talk a little bit about RNA sequencing libraries now. And for extra cellular vehicles, like it all depends on like, the RNA quality, what we are getting from those, and because the entire RNA sequencing platform, whether it is, I'm talking about the bulk sequencing here, it is a poly-A tail based. So if there is poly-A tail, then it is okay. It could be total RNA or mRNA, we does not matter. But if it is non-polyadenylated RNA, then there is a suggestion or protocol for addition of all the poly-A tailing. Because the chemistry is based on, some oligonucleotides, which has DT sequences, which are complementary to the poly-A tailing. And if there is no poly-A tailing, that's why the entire chemistry is not going to work. So I think that is I would say, the limiting factor here, when we think about the RNA sequencing. Okay, I am gonna, talk a little bit more about RNA sequencing, and that time, if you have any additional question, I'm happy to answer.
Can we polyadenylate it on the small coding, not coding region, and then sequence it?
Yeah, that's what I meant by internet polyadenics. So we do have some, protocol, but okay, let's go to the RNA sequencing slide, then we can talk more about it. So when we think about library preparation in terms for Oxford Nanopore, so we have, output optimized library, which you did yesterday, and the chemistry is called ligation sequencing. And it comes with, like, if you can do it as a single plex or multiplex, and yesterday we did the multiplexing. We also have speed optimized, which is called rapid sequencing kit. The chemistry is different. Chemistry is transposes based chemistry. It also comes in, it has also option of single plex or multiplex. We do have chemistry for ultra long reads. And this is very relevant for de novo assembly projects when we are doing entire genome assembly. So that kit is called ultra long DNA kit. If if you're ever interested in de novo assembly, I'm happy to help regarding this. The chemistry is transposes best, but it is more about the previous steps, like how we are extracting the high molecular weight DNA, because we don't want to shear the DNA if we want to do ultra long reads. There is low input optimized, when we don't have enough starting material. It has the PCR step. There is targeted sequencing for specifically for the microbiology or microbiome space called 16S barcoding kit. And for RNA, we do have direct RNA sequencing kit where there is no need to do reverse transcription. You can read the RNA strand directly, but there is also cDNA-based approach where you can utilize reverse transcription and then make a library out of it. Also, our platform is compatible with 10x, Chromium, and then the kit you use is similar, like ligation sequencing kit. So the cDNA needs to be done using 10x Chromium, either 5' or 3', or special, and also will soon publish protocol for TCR-DCR that is also working well. And for the RNA also, one thing is there is multiplexing option available now. I mean, it is gonna come out in January.
So when you say compatible with 10x, that means making libraries with 10x and then loading it onto P2 Solo?
So cDNA needs to be done using the 10x protocol.
Okay.
And then you have to do the addition of the motor enzyme for doing it in Nanopore platform.
Okay.
Yeah, so the 10x part, for 10x, if you want to do the short read sequencing, is the same thing, right? You are doing cDNA, then you are fragmenting it, and then you are following the steps for making it compatible with the Illumina platform. So it is similar here.
Okay.
You are doing cDNA, and then you are making it compatible for the Nanopore platform. So let's talk a little bit about the chemistry now. So we have done this ligation sequencing yesterday. So the way, it works, I mean, if you do single plex, so the first step is like, end repair or nick repair, and this enzymatic step. And then you are adding the sequencing adapter. Fragmentation is optional. It depends on the target of the project. And when we are talking about fragmentation, we are talking about in the gDNA context, because sometimes gDNAs are really, really long. If it is too long, there is a tendency of pore blocking. So then we have to, think about which one is better, like maybe a fragmentation, a little bit smaller size will give us better data. So this is not relevant for cfDNA because cfDNA is anyway way smaller than gDNA. Yesterday with this thing, native barcoding kit, and we already went through all these steps. So we started with cfDNA example, and then we have done the end repair using FFPE enzyme, and then we added the barcodes, like ligated barcode, and then ligated the sequencing adapter. And it says 140 minutes, but it is way longer than that. So I don't think this time is correct here. So the speed optimized kit, which I mentioned earlier, it is called the ligation free kit or rapid sequencing kit. The chemistry is transposon based. This is like rapid means it's literally you can do all the library prep within like 20 minutes. So the transposon complex already has barcode. It does the tagmentation and attach, if the barcoding is needed, attach the barcodes. And then, there is the attachment of the sequencing adapter, which is similar for all the chemistry, and then you load samples. So this is good. It works perfectly for like, it depends on what type of application you are doing in your lab. Now let's talk about RNA library preparation and multiplexing. So the RNA flow cell, which I mentioned at the beginning, you can do this chemistry. Or RNA flow cells are only applicable when we are thinking about direct, sequencing the native RNA strand. The way this entire chemistry works is like it has to be polyadenylated RNA. So it could be total RNA. If total RNA, then we have to enrich the polyadenylated RNA, and we have suggestions how we can do that, or like purified mRNA. If there is no RNA, then no polyethylene, then we need to add polyethylene enzymatically using polyadenylation enzyme. So there is a primer annealing step, and the primer, as you can see here, it has T poly-DT sequences, which is complementary to this region here, and that is the reason, the polyadenylation is necessary for the RNA-based chemistry. Although, we don't, reverse transcription is not, it is an optional step here. But if we do reverse transcription and we suggest to do it, then what's happening, this entire thing is acting as a scaffold, and then you can avoid the secondary structures of the RNA. Which, if there are let's say multiple secondary structures or complex structures, which are very common, right? Then it might block or hinder the entire, let's say, motor enzyme. So just in order to avoid that, the reverse transcription is recommended. But when you are attaching the adapter, it is going to only read the RNA. It's not reading the cDNA. So, and that is the reason it is the direct RNA. So even if you do reverse transcription,
Okay.
the cDNA is only acting as a scaffolding platform, and we are not sequencing it. This entire RNA sequencing for the direct RNA platform, it happens in 3' to 5' direction, but the software actually reorients it. So the data you are going to get is in the correct format, like 5' to 3'. A correct format means that's what conventionally we do, right? So there is also option of reverse transcription based kit where you are doing the reverse transcription. There is UMI incorporation happens, and then there is some kind of PCR amplification, and you are attaching once again the motor enzyme and sequencing it using DNA flow cell because this is cDNA based approach. So this one already has multiplexing capability. So if you are not, looking for direct RNA, this platform is great. So what is the extra information you get if you do direct RNA? You can calculate the detailing. I think the only extra information here you are going to obtain is the RNA-based modifications, which you are not going to get if you do the cDNA-based approach.
So, suppose you are looking at, I think Terry might have asked you this yesterday and I was not paying attention. The microRNAs, they are very small.
Yeah.
And they can you, they are not polyadenylated. So can we polyadenylate it and then use the direct RNA sequencing?
I think,
or is it too small?
It is I think, so first of all, I would say I don't think we have any benchmarking data for microRNA sequencing to support anything. But I know that few of the labs are trying to optimize it. If you are really interested, I can explore it. But my understanding is, so theoretically it is possible. You can do polyadenylation and you can use this platform. But sometimes it also depends on the length of how many A's are getting added there.
Okay.
And whether the software will be able to base call it properly. So I am not sure it would be a very optimum platform for doing microRNA sequencing. I don't think.
What if I make the library using a different kit and then add the adapter?
I don't know what you mean by different kits.
Like, so I did go to Illumina and ask them if there is any way we can make library using library for microRNA. So they don't have a good library making kit for microRNA, but they did recommend a different company. I think it was Kiagen, and even if I have a cDNA library for these microRNA, can I run them on?
So I need to know exactly, what are the sequences there, what it would be, whether it would be compatible or not. So in my past research life, I have done a lot of small RNA sequencing because I used to do ribosome profiling all the time.
Oh, nice.
So there are, with the small RNA, I think the issue is sometimes polyadenylation.
Yes.
It is very hard to sometimes maintain how many A's are getting added.
Okay.
So there are like several different troubleshooting you might need to do if you are interested to explore the small RNA sequencing, specifically using this platform. And my question would be like, what extra information you are trying to get using Oxford Nanopore, right? So here, like, the information you get using our platform, which you absolutely miss using short read sequencing is like example isoform level information. You are not going to get it there. Also the accurate polyethylene, you are not going to get there. And then even the transcript abundance, like you are getting it for really, long, like end-to-end information, which is not possible using Illumina. We miss a lot. But for small RNA, I think let me do some internal research because I think the issue is one is polyadenylation because that is my experience from all the ribosome profiling I have done in the past. We used to do it using template switching. We're using Takara. That is actually probably the best way to do that. But for Nanopore, I need to just check if some lab has already optimized anything for the microRNA, then we can talk more. Yeah, we do not have any present benchmarking data. Okay, so I think let's talk about CFDNA now. And even if you don't do CFDNA for any kind of gDNA, extraction is very important for any kind of genomics platform. So clean out the sample, you get a better data. So what I understood from all the extraction method you have done for CSS, CFDNA, it looked like there is abundance of high molecular weight DNA. So now we don't know whether those are gDNA or something else because, from this fragment analysis what we have done, I don't think, first of all, it was not resolved properly using that test station, and it's hard to know. But in case, let's say that is all, gDNA molecules. So I think yesterday we discussed the next thing which you can try is double-sided bit cleanup and try to make it more clean, and so that we have more abundance of the CFDNA, like mononucleosome, dinucleosome peaks, and not like really high molecular weight peaks. So I think that way probably we can achieve more coverage if like our guess of those high molecular weight peaks are actually gDNA. It's kind of hard to analyze it from there. So these are the very standard QC measurements we suggest to do, and please do it all the time because these are very important. One is for any kind of quantification, it should be qubit. The reason is qubit is a fluorescence-based method, not absorbance-based method. And as you know, example in nanodrop, because it is an absorbance-based method, it is also giving us the signals of any kind of free nucleotides, any kind of ATP or any kind of NTPs are present there. So for quantification, that's why any fluorescence-based method works, like it is more accurate, more sensitive. So double-stranded DNA as a kit is like recommended for most of the samples. It depends like what type of sample we are dealing with, but this is what would be my suggestion. For nanodrop, it is also a good measurement because it tells us a lot about, what are the contaminants present, right? Example, if there is hydroxyl group present because of the presence of ethanol, isopropanol, or like EDTA contamination or any kind of salt like vodinium chloride or isocyanate, phenol. Sometimes it happens, right, because of the purification, whatever we are using. So the ratio up to 60-80 to 60-30, it's a good, I would say, like a quality check metric. It does not tell us everything about all the contaminants, but it's a good way to check purity. That's what I can suggest. Definitely, assessment of DNA fragment length is required. It can be done using femtopulse, which is probably the most accurate in my experience, but it is not available everywhere. If station or bioanalyzer is a good alternative, or like state of the art gel running could be done as well. But it is very important to assess the fragment length distribution before we move to the library preparation step. Storing library for a short term 4 degree. For long term, I think -80, that's what recommended from Nanopore. We already talked about cfDNA. I think this extraction method is quite specific for the plasma, not for the CSF. Yesterday I was checking, different papers. It was a very quick research from my side. I would do more. I found a bioarchive paper. I think it got submitted in 2025. Check their extraction method. It is completely different, and the way also they made the library, they did not follow our suggestion. They have kind of optimized it.
Oh, yeah.
Yeah.
Yeah, we should find it.
But then finally they added the way they did it, like after barcoding, I think from barcoding step, and they followed Nanopore protocol, but prior to that they optimized their protocol. So the data quality looked actually good, so you can check that paper and it might be useful. Yeah.
I mean, what if like the nanodrop looks perfect, like it has the proper peak, but the tape station doesn't look good. So which one do we?
So those are for nanodrop is giving us a different information, and tape station is giving us an information, right? Nanodrop looks perfect. It only means like only the contaminants which we can talk about from the nanodrop. Those contaminants are not present, but it does not say anything about the fragment distribution. So both has a different purpose, so it's not like a complementary method.
Yeah. Okay.
Yeah.
Okay. I'll have to, but I'll be back at here.
Okay. Yeah. So yeah, I would probably quickly go over accuracy now, and then I'll stop, and then we can again talk in the afternoon. We can just yeah, no worries. So for Nanopore, like the chemistry we have is Q20 plus chemistry. So presently, the accuracy we have in the SUF model is Q26. So let me actually talk about the base calling model briefly now. I know those slides are coming later, but when we do the base calling, there are three types or three modes of base calling. One is fast mode, which is the lowest accuracy. One is hack mode, which we did yesterday, and another is super accuracy mode, or it is called SOUP. So if we use SOUP, it is Q26 plus raw read accuracy, which is 99.75%. And once again, I want to mention this is raw read accuracy, not consensus accuracy, which you see in other platforms. So for Nanopore, we also share the raw read accuracy, which means like information of each nucleotide and at what confidence level or at what is the Q value for calling each basis. This information is not available in other platforms. Example, other longer platform like CatBio, the interchemistry is consensus. So they're reading the same strand again and again, and then you are getting the consensus read accuracy. So it is not like, I know like some scientists like to compare it, but we cannot compare because those two are very different metric. So presently the Nanopore we have, it's called R10 chemistry. So it used to be R9, and there have been improvements, and the improvement happened because now like the length of this protein Nanopore, it changed. It is now increased. And the way the base calling happens, as well as the trained model is different now. We have more information, so that because of the combination of these two factors, the raw read accuracy got improved a lot in the past year. So the base calling, this is what we have done yesterday. It's called high accuracy base calling model. So for cfDNA, this hack model would be sufficient, in my opinion. And it always what base calling model we want to use. It depends on what data or what type of variant calling we want to do. Example, you can see here the difference, for, let's say for methylation calling. If you use hack, it is 98.8%. If you use SOUP, it is 98.8%. So there is no difference. For SNP calling, there is no difference. Now, if someone is interested in indel, then there is some difference. And that time, I think re-base calling using the SOUP model will benefit us. Otherwise, hack is actually probably quite okay for doing all the cfDNA-based sequencing. So the modification calling, there is a modification calling button whenever you want to do methylation. We need to just click this so that the modification callings are on. Then we have to check, we select here what type of modification we want to call, or at what context we want to call. So there are options based on our requirement. We need to select that.
So just to clarify with kind of this box, this box is all for base calling off of the squiggle, right? Or at 50 seconds, the electrical current.
Yeah.
We also have that electrical current data saved as well, meaning all of this can be post hoc reran. Okay.
So you can rerun, and as you mentioned, because yesterday we did not do the base calling, so I checked it. The post-run base calling, let's say we want to do it. That time you can do base modification calling as well.
Right. So we have to save squiggle, and all of this is if I forget to do something, I can do it later.
You can just re-base call. Yeah. So for that, the software is called Dorado. We need to do the standalone Dorado, and I think tomorrow, let's see how the run goes. I would like to install that as well, so that at least, just for learning purpose, let's do one Dorado
Yeah.
this calling, using the modification on, then, you know how to do it. And then you can just play with the data. You can use all the workflows which you think would be best suited for your project. And, you can play with that data. So I'll try to do it tomorrow. And I hope the run would be mostly over by tomorrow. So this is the modified base model we have, and I think this is the latest table. So for DNA, and let's only talk about DNA now. So you can do it in CPJ context or like in all like base context. So 5-MC or hydroxymethyl calling is possible, 6-NA is possible, 4-MC, and that is not actually relevant for you. It's possible in 6A. Yeah, it is possible. And Dorado is the post-base calling, post-run base calling. So you can do everything here. And this is, this is like more relevant for the whole genome sequencing. And they are comparing because state-of-the-art method was using bisulfite method for methylation. And using Oxford Nanopore, even with 20x coverage, you are getting way better data than this. And if you do 40x, I think it's quite perfect. So methylation calling, it's working actually quite well. All the labs, especially all the oncology labs I work with, they do it quite often. So it's working very well. Yeah. So tomorrow, I will talk more about the specific to the methylation calling, the workflow and everything. So I think, I think for now, let's stop here. And later I am gonna do, let's see what I have here.
Hopefully it's cell-free as little GPNA as possible.
Yeah, I sorry to interrupt. I don't think I need to talk about hardware check because you already did that. I will talk a little bit about the flow cell, like, how the actually the sequencing is happening, like, how many ports are open or how many channels are open, just to have that idea. And more about the sequencing report, how we can analyze, like interpret the report, and then a little bit about the epitome. I think that's what we can do today. This is like how the flow cell is. It has like wells and electrode. And for PromethION, okay, so an each well so has like its own, like every well has its own electrode and contains one pore. And in PromethION, there are like 675 sensors or channels. So it is a little complex, but each channel is connected to 4 wells. So here it is showing, I think this cartoon is probably better. So this is like one channel, if you can imagine this like imaginary line. So these are all channels. So what happens during mock scan? We kind of like the Mino software, it kind of tests each well in the channel and select just one well for doing the mock scan. It's not like even, when we are doing the flow cell check, let's say we have 8,000 active pores. It's not that 8,000 active pores would be sequencing at the same time. Okay, so this is how basically works. Now, I mean, I don't want to overwhelm with this wash and like reload part because I don't think for CFDNA it is relevant. But if you are dealing with any other type of DNA sequencing in future, this is like we can always recover more pores which are inactive and then reload samples. In that way, we can get more data, more gigabases. But for CFDNA, because of the small sizes, we don't need to do it.
Could you speak briefly to, I guess, do we have these cells that are running currently? Can we still wash them and put them back in the fridge and reuse them later, or are they done after this? Like how
You
that
can
works?
do that. So there is a wash and reload kit.
Yeah.
You have it, right?
Correct.
Can always wash it, and then I think it has the recommendation like how to store, like it should be in a buffer. So what buffer it is, I think the protocol has a recommendation. I can also share the protocol, and then you can keep it at 4 degrees, and before you keep, you can check how many pores you recovered. Let's say now it has, I don't know, 100 pores. It started at 8,000. You will never recover 8,000, but even if you recover 50%, it is good. It is good for, I would say test run, because the reason is you have used barcode, right? So if you want to do barcoding, maybe I won't use the same barcode.
Okay.
Just to be on the safe side. There should not be mixing. I haven't seen it, but I'm just to be on the safe side. Maybe you have used 1, 2, 3. Let's use like 6, 7, 8 and run. But in clinical lab, I know it is not a clinical lab. We never do that. That's never we never do that. But I think for research purpose, especially for pilot, I think that's a good idea.
Okay.
Yeah, I can share. I'll share that protocol. Okay. Now I would directly go to monitoring a sequencing run. We already talked a little bit about it. Go to experiments part, right? And then you would be able to see how many samples are sequencing. And then if you double click here, you would be able to see all the channel panel. And also there is an option of export run report, which we have done in the morning, and where you get all the run related metric. So these are the 3 I feel like there are multiple important parameters, but I think it's always I check pore activity and pore scan. So it tells us like, how many, let's say here, pore scan result. It's not. So how many, active wells are there at the beginning, how it is declining, and different other metrics like what is the active or available pore versus saturated, unavailable, 0 or inactive pores. Same thing like, this is also percentage of pore activity. I feel like these are kind of complementary, right? But the metric or the y-axis is quite different here. So I'll go over. I have sent an email where we have more description of what individual pore characteristics of like this metrics are. I'm gonna go over that. But what else is there? So this is the channels panel, like which you see as well as in the sequencing run. I think where we have to see here once again, we are trying to see like how many pores are actively sequencing versus reserved and inactive. Like example here at the bottom, right? Here. If we see like a really huge number of inactive channels like this, that means like at the beginning, it could be indication of something, air bubble or something. Sometimes, if you take the flow cell, you will be able to see if there is a big air bubble that is kind of visible. Otherwise, here also you would be able to see. I mean, for us, it did not happen. It was quite happy yesterday. Now, you see more like inactive, it is because the sequencing is done. There are not probably DNA strands left because we did not overload the sample. We kind of underloaded, I believe. So that happens. Most of the sequencing is already over. So this is an example of good library. It's like lots of pores are active, sequencing, very few numbers of inactive pore. Very less in a saturated, very less multiple. I mean, so the green color portion is higher here. So this is a good example. So pore activity. So this is now what's happening, right? It started with really high pore occupancy, and slowly it is decreasing. So what is the percentage of pore occupancy at the beginning? It is kind of an important parameter, and it depends on whether we are loading it properly. If we are not load it, then it is gonna, there would be sharp decrease as well. So underloading, we'll try to avoid underloading. What else? Yeah, so this is also kind of, I'll show again. So for our samples, we don't have that much of adapter contamination. During the library prep, if, for some reason, the stoichiometric ratio between the adapter protein, like adapter and the DNA molecules on which we are adding that, if it is messed up, then you might see a lot of adapter contamination, which is the yellow colored part. And that will just decrease the reads what we want for our sequencing, because you will be just sequencing these adapter molecules then. But for our run, I think what else we can? Yeah, so this is going to happen. So for this one, see, it happened after like 1 day, 20 hours. So this, the blue part, which is the inactive or zero pores, like available, it happened like almost after close to like 2 days. So I think that's not bad, at least for CFDNA, that's what it looks like. Sometimes what happens if you see a sharp drop in the pore in like 4 or 5 hours, if it happens, just pause it. Try to probably take out samples, reload and see, like purify and reload and see if you can, if you're seeing more pores, or just transfer it into different, so that's also possible. Yeah. See if it happens, you can let me know, I can suggest something.
So we don't let it finish, we just pause it?
There is like, let's say after, within 2-3 hours, there is a sharp pore drop, that means there is something wrong. It could be because of the flow cell, it could be because of the sample. So I think it's always a good idea to recover, purify, and then reload in a different flow cell. Or there is also a protocol for that. How we can retrieve the samples, I can share that as well. I think that's also something important. Yeah, so sometimes, if you see like this is although this is showing maybe this one, within few minutes you see there is a huge pore drop happened. It is usually because there is some kind of contaminants present in the sample, which is blocking the pore, and the pores are not able to deal with it. So that indicates the sample needs purification. So that it is, if it happens, I think that would be the suggestion. So this is another one where like within 17 hours or so, you can see there is huge amount of the blue part, which is the inactive or zero pores. It can happen because of the increase in membrane stress due to salt concentration or some kind of pH imbalance or presence of surfactant. I think in this case also, because the salt is coming out of the sample, the suggestion would be purify the sample more. So yeah, so if there is any kind of osmotic imbalance happening because of contaminants, you would see this kind of phenomena. And I think this one I was kind of showing earlier. We have the read length histogram, we have translocation speed, barcode, distribution, and the quality score. These are other, parameters which you would see in the run report. And I think all good kind of a QC to understand whether the sequencing is working well or not.
That read length distribution was for GDNA?
Yeah.
Okay, good
This is
stuff.
not for the CFDNA. CFDNA is like I checked other reports. Looks like what you are saying. So I think it was okay. Only thing I felt like it was probably a little bit underloaded. That's the only thing. We can improve that, but otherwise I think the run looked okay. Yeah, I think stopping a run, these are kind of an important thing because if we ever want to stop the run, there is a button. You can stop the run, and then it asks you whether you want to stop sequencing and base calling or only stop sequencing. If you do the stop sequencing, then the device will finish base calling and it will generate the FASTQ, because there are two things you saw, like one is estimated and another is whether best call already the best calling is done. So there is a lag between the estimated and the real-time best calling. So if you ever want to stop, I feel like this option is safer. Like at least the leftover base calling would be done. That's what I like to do. Yeah, so if you stop sequencing, then you will have to start the run for the flow cell from the beginning. What else? Yeah, I think for today, this is what I wanted to share. I would also share the email I have. Actually, I have this. Yeah, absolutely.
So when you use a 1 ml iPad, the sample in the site, you do like this. You use your finger to pull it. So I'm thinking about the 1 ml, the tip, just a touch, or you can another press or another loose.
No, I was just touching it.
Just touch it. That's okay.
Yeah,
that's okay.
So only thing, there is no perfect protocol for that.
Yeah. Your
How I
experiments.
feel it,
Yeah.
like this is working best for me. And so the purpose is like it should not introduce an air bubble.
Yeah, that's why we were very worried about the air bubble. So you can do like this, you can do like this, then you still have another one drop here, and you just take off like this.
Yeah.
Or when you do it straight, you do that, or this.
Yeah, you
Another
can
angle.
If it is, I
That's
don't
okay.
think that's okay. I don't think it is going to change anything. Yeah.
Okay.
So this is quickly about all the, the pore scan different parameters. I think the most important is like the reserved core. What does it mean? So when the mark scan is happening, the voltage is close to 180 millivolt. And what we know the software is doing, it has to decide, right, whether that pore is, let's say, good or bad. That's what it has to decide. So what it does, it measures the signal for 10 seconds, and then it classifies each time point as one of the several signal states. Like, so if at least within 2.5 seconds, in the Mino software, it can get a good sequencing data, and then it classifies that pore as a open pore. So this is kind of a selection logic, and I feel like this is a good explanation. It's like a battery life, So you remember like the figure I showed, like the channels has like four pores, and it just only open one. So the way Mino does it, it has a ranking system. So it ranks the 4 wells in each channel based on another metric it calculates. It's called mediator drift. So we don't need to remember everything, but this is how the logic in the algorithm works. And the mediator drift is kind of like a battery life. So it selects the best one for the open pore, and the rest those are the reserved pore, like a backup pore. So the best one will participate in the mark scan first, and the rest would be like the backup pores. And when it does the mark scan next time, it is going to select one of those pores. So this is how it works. So when you see saturated, it means like the membrane is gone. So there is too much electrical energy passing through it. There is a very low resistance, so it's not, they are not going to use that. What else is important? I think reserve pore, the unavailable one. So this is also same non-functional one. So it means like the sequencing is not happening, and it could be because of some contaminant there. So those pores are not unavailable, and there is another one which is called zero. So there is no current for the zero pore. So typically, it also means the same thing. It's not available for sequencing. So I think to understand the run report, the main thing is the singular reserved pore. Yeah. So I think this is all about the Mino, how it works and everything. And we can now check one of the run reports from yesterday's run and see how does it look like.
All right, so this is the CSF report.
Okay. Yeah, so we can go over it. So let's see how much data is generated for this one. So the reads generated are not that much. It is close to 40 millions only. I mean, okay, yeah, but it's only close to 20 hours as well. That's probably what we expect. Okay, let's move on to the.
So this is where we would be turning on the methylation.
Methylation, yeah. Everything else should be okay. It's only when we are starting the sequencing, there was a actually tab. It says methylation on or off. There you will do the on.
Was it on during the first ones or not?
It was not on.
Okay.
However, we can decall post hoc with methylation. So it sounds like all to say we're not missing the methylation data. We just won't have real-time calling on it.
Oh, there's real time part. Okay.
Yeah.
Fine. All right.
As far as I understand, I was worried for a moment that we would not be able to do that.
So you can do it. And also, not all the methylation calling can be done real time.
Right.
So for some methylation call, we always need to do it Dorado standalone anyway. But I think they are going to include it. That's what my understanding from today's meeting. So it would be very comprehensive. Even whatever is missing should be part of the Mino soon. And what else? Yeah, that's what about methylation calling. Yeah, so port 5 needs to be, there was an option, right? Remember, like whether you want to generate port 5 or not. Definitely generate port 5 because we want to do the methylation calling. If that is missing, then I think we cannot do it.
I think right here, 1 file per hour.
Yeah, we actually
We did it.
yeah, we did it. Okay, so here it looks like this is
I missed something, sorry.
Yeah, sure.
Wait. The benefit, I understand the benefit from yesterday, it was real time, to see how things going. Yeah. Could you summarize the benefit of looking at the real time methylation calling versus, is it like a evaluation of how well things are going? Maybe you can see that the sequencing is progressing. We have confidence that it's going well. Like what, for me, the bottom line, like why would I want to see the real time methylation?
No, we want to see it. Yesterday, I just did not do it because, this is the first time, we are doing the pilot and we can do it as a post-run based calling. But I think it is always beneficial to do it real time. Only there are few methylation call. I think on humans, I think only one, which cannot be done using Mino. For that, you need to do it using Dorado. Which one is that? Yeah, so Dorado is the post-run based calling software. I think there is only one, if I remember from the table.
I understand one benefit is just that you get all the data. Like the time that it takes sequencing, it's also base calling in the background.
Yeah.
You just can do analysis after your sequencing is done.
Yeah.
Whereas now we're going to have to wait 3 more days.
And also, it is longer now.
So even 5 more days then.
So whenever we do it using the standalone Dorado, it is more time consuming.
So this is like a consideration for like rapid field sequencing in particular.
Sorry.
Like if you're doing not this experiment, but like doing like field sequencing, and you need to do it rapidly, this would be like a consideration for that.
Sequencing, for the Minion, that's what you mean? Or using the P2 Solo, the same platform. So if we use the P2 Solo, it's fine. You are getting real-time data, including the methylation. If you use the smaller platform, I think it would be also hard because those are not that computationally. This is computationally very expensive process, right? So that is maybe not possible as well, not in all contexts. Yeah.
Thank you.
So clinically too, there would be some benefit if you are using it for diagnostic or.
Yeah, that's what I'm not getting at. Like how quickly you needed it.
Yeah.
That turnaround time. But for like our plan analysis, it can happen. For the educational benefit now turning it on. But like our plan, we're not doing diagnostic.
Right. So I think we're okay using this data still and moving forward. We can just rebase call it.
Thank you for recapping.
No, that's a good question actually. So now read length distribution. So I'll show some other data. And it looks actually correct, even other labs got the very similar distributions. I think it looks okay. So the thing is, since I don't have any CSF run report to compare, I cannot talk a lot about it, but it looks very similar to what we expect, right? 3 distinct peaks or 3 distinct distribution because of the different nucleosome like mononucleosome, dinucleosome, and polynucleosome. So it looks, I think, good to me. Yeah. Let's go to the next. Yeah, so maybe this is, so we can skip, maybe the reads one. The barcode distribution also 1 to 3. I think it looks okay. That's how you distributed it, right? Percentage of bases.
Yeah, I think there's a table that
Also gives that, yeah. So that is also always, check how many reads passed versus failed. The Q-score histogram. It also tells us what is the median Q-score we are getting. And I think it is very comparable to what we see in cfDNA. So I think it's good. This is good, all stable. Like it is the Q-score once again. High-wise, there is no drop or anything. Here, just I'll check, uncheck the grouped so that we can see the adapter distribution or many more, parameters. And as I was describing, the main thing is how many ports are available, how many ports are actively sequencing. And you can see there is no pore or it started to get increased because it's also almost close to 20 hours or so. This is what we should expect as well. So it's fine.
What is this like every hour downshift? It's where we see like a decrease in sequencing occurring here.
Yeah, so it's every 1.5 hours there or 1 hour there is a mark scan happening.
What? I'm sorry.
So the mark scan, like the Mino is, checking which ports are active and which should be the reserved pore. So that phenomenon is called mark scan. So that's how we set it up. I think we can do it every 1.5 hours. That is also okay. I think yesterday, I remember when we were doing it, by default it was 1 hour, so I did not change it. I think that should be okay as well, right? Here also the same thing. Yeah. So if you see like a sharp drop here of active pores or which like available pores, then that is concerning. Like in 5 hours, you see like 90% is gone. Then something is, there is, usually it is because of the sample contamination. Because there is osmotic imbalances happening, electrical, signal is getting like a lot of noise there. There is very low resistance, all the things which we talked about earlier. So, but for this one, it looks okay. Translocation speed looks stable, temperature looks stable, so which is a good thing. Yeah, I think, yeah, so that's all it is, yeah. Let me quickly show you. Then, boards, I'm gonna show just reports. Yeah. Let me share. Yeah, this is, one of the cfDNA sequencing report I obtained from a different lab. I do not know, I mean, it is not CSF, I know it is plasma. So I don't know, I don't have any other biological information available, but, I think it is quite identical to what you are saying, like kind of 3 peaks, right?
Can we go up to just start at the top with the amount that they sequenced? Get a perspective of.
Yeah, I can probably share this.
Okay.
This report to you. Okay, so here I think this is a good thing like, they did the base calling, like methylation on, then they also did a soup base calling, although our kind of protocol which we followed, it says hack, but they did, so that's okay, that's fine. And yeah, so you would be able to see here what's happening, and these are the parameters they have used. So maybe I'll just share it to you so that when you do it next time,
Right.
use the same thing. Yeah. What else is there? So the barcode distribution, I think I've used so many barcodes. The Q score looks quite similar to the median, which is a good thing. So they are, I think they also overloaded the samples, which is a good thing for CFDNA. That's what when I was inquiring today. So the NYU lab, apparently they always overload it and they just let it run for 72 hours. So if your sample is not limited, try that, because they are getting very high coverage with that. So yeah, so you see like the active 4, I mean, it was at least like, I'd say if this is 60, it became like 30 here. Or, I mean, they were pretty good till 30 hours, I would say. After that, it slowly died, but I think that's what you expect.
What happened at the end? The pores get activated again, or?
Yeah, so the pores are now, so this blue is probably no pore. So no pore is like, there is no electrical and like a signal getting detected. So these are, I think this is the color, right? Sometimes it's very, I think they should change the color because, yeah, I think this blue is this blue. So this is the no pore. And the 0 is this here. Yeah. So, the 0 means like completely like no current. And I think no pore is like unavailable is the contamination. Let me see. They have the distinction of no pore, but here. So yeah, it's just if the marks can fail to detect any pore. I think it's because like mostly unavailable. It's all used up because of that. Yeah, so it is a maximum 0 pore. So no electrical current detected. So it's all dead that time. Yeah, I think this is one. Another one, I think it's a little bit different. It has more like, still it is 280. That's the median. And they also kind of used 3 barcodes only, like the way we did, so which is a good thing.
So
This is not ours, right? No. So it looks so identical. Yeah. So they ran it for 72 hours, but you see, like, it got decreased within a day. So this group, whoever did this, did not overload the sample. Yeah, I don't think this is like an ideal run because it looks like it's even after a few hours, this green part unavailable got increased, which means the sample had some contaminant. So this is not an ideal run. Although their distribution was similar, but this is not an ideal run. Yeah, I won't. I think your run looks way better than this. So this is another one, similar in 50, 3 peaks. They did hack, modified base calling was off. How many samples they ran here? Just one. I don't see anything else.
maybe the next page.
This is 24. Okay, maybe you're right. Okay, no, 3. 3. 15. That's the median Q.
Very similar to our plasma.
Yeah, very similar. Let's see. I think this looks a little bit better than the previous one.
Yeah, a lot.
Right? Because the previous one, like this
Down.
went down really quick, but still it also has quite a good amount of like something is there, because that's why it got decreased so quickly.
And which color are we looking at for indicators of contamination?
So I think this is the unavailable one. That's what this metric, it's here. Oh, I'm not showing it here. So unavailable is the most common non-functional pore classification, and it means something is preventing the sequencing, so which is this color. And it is usually because something is there. It could be salt or maybe wrong pH, some other contaminant. Hard to say what. Something is blocking the pore, but this color, not the 0. 0 is like no current, completely dead. The translocation speed looks good, but at the end it dropped. That's what you expect. I think that's what it is. Yeah. So from all of this, I think what we can say is, I think for you only suggestion which I have presently is can increase the input and maybe for one sample you can try doing the double-sided bit cleanup, and if maybe it will improve the quality, but I think we would know more about it when we do the data analysis, whether we have vDNA contamination or not. If we don't have it, then probably we don't need to do even double-sided. Yeah, so I think that's what at least my analysis is for today's run.
Today, I'm gonna start with the data analysis part, how we can handle all the files which are generating out of our sequencing platform. For your context, it is P2 Solo, and the data you will be dealing with mostly, or in the recent future, would be cfDNA sequencing related data. So as you already know, different tools, might need different file formats, sometimes needing file conversion, so sometimes bioinformatics software, as a result of a research project, can be might lack support. So that's why most of our solutions are quite well maintained and fully supported. And first, I think for the data analysis part, I want to directly go into the different file formats which you will be dealing with when you are doing Nanopore sequencing. So Nanopore raw data has traditionally been stored in fast file format. That used to be the case with all the Nanopore data. But presently we have a different format which has some additional benefits. Example, reduced file size, saving on storage, and also requires less computational resources for writing and readings and saving. And this file format is called POT5 format. So this one, POT5 stands for Pore Open Data Format. And whenever we are sequencing using Mino, so this is the file format you will be obtained for all of our sequencing run. We should also know a little bit about another file format which are very common across I would say most of the genomics platforms, it is called FASTQ file format. So this is kind of a de facto format for most of the sequencing data types. This is a text file, quite large in size. And for Oxford Nanopore specifically, this is how the FASTQ file should look like. Example, it has a header, then sequence. Again, header should start with a plus sign, and then you are going to get all the information. Everything is embedded in this file, starting for like from base quality, all the base specific information. And it is, the Q score here is represented as ASCII characters, like American Standard Code characters. So, and all the other information, starting from read ID, run ID, sample ID, everything should include in this text file format called FASTQ. Okay, so we also have added option to configure how long each group runs for. So the default is 8, but you can either increase or decrease that. And this is like a table. It is showing the gigabases output you are going to get out of all the flow cell. What is the size of the POT5 versus the zipped format of the FASTQ or the BAM file. So this is, I feel, like a good information to have for size of different file formats. So here we are showing, like, as you can see here, like the POT5, which is the Oxford Nanopore format. So the data is shown in Arrow library. It contains the raw signal data, metadata, and because of that, we have the ability to do the rebase calling using the standalone Dorado software, which we might try today. But this is like size is larger, and presently, and if you remember when we are starting the sequencing run, day before yesterday, we need to make sure that the POT5 format is on when we are starting the sequencing run. So, and I think this is quite important because if we want to rebase call it, then we need this POT5 like files. And the BAM file is more like a standard format, and I think you already encountered it because you are dealing with short-read sequencing samples. So this is the alignment format. And for us, like the BAM, like it is also storing the modified base callings. It is very, I think, important for your work because you would be doing methylation calling a lot for the cfDNA sequencing. This is also a text file, text-based format. It's smaller in size compared to POT5. And this is a default format for all the modified bases. Okay, I think I'm gonna skip this. So Oxford Nanopore not only provides sequencing solutions, it also we provide a wide range of tools for data analysis, from base calling to QC, a variety of analysis and reporting. So in this table, we are seeing a list of software and which stages of the analysis they cover, and what level of expertise is required and which kind of support we provide. The software, for us, in order to understand the analysis part, I think it's most important is like we should know that the software that controls the sequencing device is called MINO, which is here. So it's controlling the sequencing, also to perform base calling, including methylation. quality control analysis of the run in real time. And there is also a limited number of real-time analysis with this software are enabled. For example, aligning to reference. There is another software which is Epitome, and Matt is going to talk more about that. So what is Epitome? Epitome is a software solution which provides a user-friendly graphical interface and access to cloud or local data analysis. Currently, I think, it is also available as a separate software. Epitome Cloud application, if available, allowed to run workflows in real time as well. It does if someone wants to use the cloud, then the internet connection is that's the only requirement. No, and it's very user friendly, like point and click. Literally, it does not need additional bioinformatics expertise. It can be also run using Epitome desktop interface through command line. So, it depends on how we want to run this. And I think Matt is going to also talk about what GitHub tools we can utilize in order to do the data analysis. Chris, I think you have a question.
So the default data, they're aligned, yes, because you say it's an optional. Sorry, I missed that. So at the end of the day, when you are running the default pipeline, we will have information about the genes and about the counts and probably the BAM files. And all these are aligned to the latest genome, the 38, I'm assuming, correct?
So that would be done using the Epitome workflow when we want to the align data, when we want to do the alignment, or we want to get the gene counts or any kind of example. Let's say you want to do the variant calling or
counts. I mean, counts per cell, similar to, for example, how it's in the 10x, in the 10x serial. So it will be, we will have to run the Epitome. This is what you are suggesting, correct?
Matt, do you want to take this question?
Yeah. So you're asking about are you asking about doing extra analyses in Minnow?
No. I'm asking about, we analyze the data to the machine, and then somehow we want the counts per cell, and we want for any gene in the skinny format or whatever, similar to what 10x provides. So what is the software that we have to run the raw data in order to know that Chris has these cells and these counts for these genes? This is the question.
Okay, and by cells, is this sorry? Is this a single cell approach?
Whatever. Sorry. So if it's not cells, if it's bulk, whatever it is.
Yeah. Minnow is not going to provide you. It's only going to give you a report on the sequencing run itself, but the Epitome workflows. And we'll talk about them in a little bit. Those are going to go over, essentially provide you with a secondary analysis. So, depending on the workflow that you're running, if it's a single cell one, it will provide you with counts per cell. If you're looking at human variation, it's going to look at, all those different ways of assessing variance.
So the Epitome. Okay, thank you.
Yeah, Epitome is probably what you're thinking, and we'll talk about that in a little bit. And if we don't cover it, then just let me know, and we can discuss it more.
Okay. Okay, I'm directly going to the base calling. So we already, learned a little bit about Mino. So Mino is once again Oxford Nanopore's devices, software that apart from managing like your device, can also perform analysis at different stages, from base calling, including methylation calls, to data analysis, including quality control steps, and in addition generates a report at the end of the run, like the sequencing report, which we are seeing at the beginning of this talk. So let's talk a little bit about the canonical base calling, which we already reviewed a few times in the past. So what's happening here in any nanopore system, the raw signal or the squiggle, like here, produced by DNA or RNA when it is passing through pore, is processed by artificial intelligence algorithms to decode the sequence of bases into FASTQ files. And the base caller is integrated within Mino, can carry out base calling live during the run, and after a run has finished, or a combination of the two. So we can choose between 3 different accuracy models, and it is dependent on the computational resources we are using with the P2 Solo, and time to answer the requirements. So there are like 3 base calling model hack, which we have used for all the cfDNA sequencing run. This is faster, and it is recommended for most applications. We also have super accuracy model, which is mostly we use for de novo assembly projects or low frequency variant analysis, and it is very slow. It takes also a long time. For P2 Solo, if we are using 2 slots, I don't think this soup would be enough based on the computational specs you have in your lab. So I would recommend, if you ever want to run soup, then the post-run rebase calling would be ideal. There is also a fast model, which is the, I would say, lowest accuracy model, but sometimes, based on the project needs, we can also use that. And when we are starting the sequencing, we have to, if we want to get the methylation calling or any kind of base modification information, we have to make sure that we are making that switch on. And that is like a toggle switch when we are selecting the Mino options, we can easily do that. So yeah, here it is showing like how we can do the methylation calling on if we are interested to learn base modification information. So this is a table where we are showing like what are the different base calling models are available, or the different versions are available. And I think for the DNA context, this is very important. It's here, we have 5MC, 5HMC, 4MC, 4MC is not probably relevant for your work. 6MA we have in the CPG context or all base context. Everything is available in the Dorado standalone version. In Mino also, I think most of the modifications are now available. When you are interested in RNA sequencing, and if you want to do the RNA sequencing base modification determination, then you can check this table. Like now we have various base modification models are included in our algorithm so that we can get the real-time information of all the modifications present in RNA. Yeah, so I think that's what I wanted to discuss regarding base calling, and a very brief overview of how the data analysis workflow works with Oxford Nanopore. And I think Matt can, give us more ideas about what is epitome and how we can utilize it specifically for the CFDNA context, if as that is very important for your work. Matt, stage is yours. You can start.
Okay. Yeah. All right. So yeah, I'm going to introduce and discuss epitome. Feel free to interrupt me or stop me if you have any questions about anything that I'm saying. Yeah, let's get started. So so our epitome software is not just the graphical interface, but it's also the collection of workflows. The main idea behind this is we wanted to make sure that we're providing our customers with powerful but also easy to use workflows. And to do that, we've wrapped them in this graphical interface and provided you with easy ways to download, manage, and configure the workflows for use with data that you get from our devices. To make it easy,
sorry.
No, it's fine. But to make it easy, we've also added local and cloud-based running of the workflows, which I'll talk more about in a minute. Yeah, you go to the next one now. Okay, all of our workflows are essentially working approximately the same way. They're all going to start with the DNA, RNA sequencing data that's output from Minnow. The software, the workflows are designed to recognize the folder structures. So if you did have a barcoded sample, it will recognize those different barcode folders and treat them as individual samples. It will pull that data into the epitome workflow, whether you're using the workflow from the graphical interface or from the command line. Either one, it will recognize those folder structures and files. If you're using the graphical interface, you do get to choose between whether you want to run it locally or in our cloud. Our cloud option will run on an AWS instance. Your data is basically uploaded to our servers, and then your configuration for the analysis run is uploaded with the data, and we will run it on one of our instances when they become available. And once the sequencing is done, you'll be able to download a report and any output files that are generated back to your local system. You could also run it locally, again, either graphically or from the command line, and you'll get the same outputs. You'll get the same HTML-based reports. You'll also get the same outputs if it's VCF files or tables of data. You'll get the same results either way. Okay. So we've been expanding the platform. It is an open platform. The workflows are open source. We do have a template workflow. So if you are, kind of all in on Nanopore sequencing, or you've developed a workflow that's very specific to Nanopore and you want to make use of our epitome platform, you can always use that template workflow or a fork of one of our other workflows as a starting place for, developing your own workflow that you can share with colleagues. Right now, we currently have, it's definitely more than 15. We've been adding a few more recently. Different workflows that cover different applications. And again, these are all available through the graphical interface or through the command line, running it through Nextflow. And again, all of the workflows require minimal setup. They all have what I usually consider to be like sort of sane default settings. So they'll work with most samples, but it's always good to familiarize yourself with all of those different options and parameters so that you do run the analysis in a way that best captures the data of your sample. The workflows, graphical interface, these are all compatible across all different OSs, so you can run these on Windows, Mac OS, or Linux. You can run it on your desktop, your laptop, workstation, an HPC, or on an AWS instance in the cloud, or really anywhere you want to run it. Yeah. Go to the next one. Okay. So here's a short list of what we are currently offering. So we have different application subjects, subject areas, like human genetics or like metagenomics. For human genetics, we have a human variation workflow, which we'll talk about in a bit more detail later on. That's for looking at germline variation for the most part. We have a somatic variation pipeline, and as the name implies, it's looking at somatic variation. So if you have a tumor or a tumor normal pair, that would be the workflow to focus on. And if you have a trio data set, you can always run it through our trio workflow. All of these workflows are going to be looking for different variants in your sample, whether they're single nucleotide variants or structural variants. And some of the workflows will also help you with the analysis of the methylation data in your sample, or looking at things like STR expansions or copy number variation. We have workflows that cover basic tasks like alignment or base calling. So these are just wrappers around our Dorado base calling software for the base calling workflow, or Minimap2 for the alignment workflow. Sometimes they are convenient to use. Minimap2 doesn't directly take BAM files, but our alignment workflow will take BAM files, and it will convert them for you so that you can align them with Minimap and then get your aligned BAM files at the end. For metagenomics, we have a metagenomics and a 16S workflows. These are both approaches. One is for more of a shotgun metagenomics approach, and one is for the targeted implicons of like the 16S, 18S, or ITS genes in your samples. For QC, we have a clone validation pipeline. So if you do work with plasmids and you want to do sequencing on your device, you can use clone validation to do a de novo assembly of your sequences, or to check for variants against your reference file that you provide. And an AAV QC that does the same thing for AAV samples. We have transcriptome sequencing. So if you're interested in bulk transcriptomes, you would look at our transcriptomes workflow. If you're interested in single cell, we support the 10x kits. You'd have to go check out the current GitHub page for the single cell workflow to see which ones are currently supported. But we do support a number of 5' and 3' kits, as well as the Visium and Visium HD kits. So we do have a lot of support there. The workflow is mainly focusing on the gene expression data. So if you do have attack-seq data or some of the other approaches, our workflow doesn't quite handle that data yet. We have an amplicon workflow. So if you do sequencing of amplicons and you want to analyze them, the workflow will align them to your references, or basically de novo assemble them into clusters and provide you with those sequences at the end. We have bacterial genomes and flu for infectious disease. The flu workflow is specifically for influenza A and B. Bacterial genomes has a number of features that work very well for isolates. So it can basically do typing for you, serotyping, or like MLST typing. But if you have a mixed group of bacteria, you can still run it through the workflow, and it can do alignments to do variant calling, or it can do de novo assembly of your genome. And then finally, if you're interested, we do have a telomere sequencing workflow, TelaSeq, which pairs with our TelaSeq protocol. And we have a PORE-C workflow if you're interested in looking at chromatin conformation. Basically, PORE-C is our version of Hi-C that you might be running on other platforms. The other thing that this slide shows, I just wanted to highlight that the different workflows have different computational requirements. So some of them with the one dot after the name of the workflow can run very well on minimal minimally spec'd devices like laptops and some desktops. But the more dots that we've added, the higher the requirements are. So anything with one, you could probably run on a laptop, maybe a small workstation. 2 dots, it would have to be a workstation or a much more powerful workstation. And 3 dots would probably be best run on like a very powerful workstation or an HPC. But yeah, just to kind of give you an idea, most of the human variation would require a more powerful computer. Same with the transcriptomics workflows, but some of the other ones like clone validation for plasmids or amplicons have fairly minimal CPU requirements. Yeah. Okay. Yeah, so if you're interested, again, all of our code is on GitHub. So these are all open source workflows. You can go into them. You can see the WF template workflow there, which is if you wanted to develop your own epitome focused workflow. These are all Nextflow. These are all Nextflow workflows. So even though it's a template for epitome, we're basically just kind of adding extra definitions to parameters, so that when you load your Nextflow workflow into the epitome software, it understands what fields are looking for. So, are they looking for a file or a folder path, or is it looking for, like a text entry, or is it kind of just a drop-down menu is appropriate? And these just basically make it easier to use it in our graphical interface. The workflows are all based on Nextflow, and all of the software that's run to do the analysis is containerized into Singularity and Docker, which helps for reproducibility across a number of different systems. Okay. For epitome, again, you can integrate, you can import your own workflows into it. I do have a few that I use fairly frequently for QC of Nanopore data. I believe there's also an alternative metagenomics approach available, the EMU package. And there's also, we've deprecated our own SARS-CoV-2 pipeline. But there is a community-developed Arctic network field bioinformatics pipeline that you can use for different analyses. I believe this one is currently focusing on mpox, but again, it's a pretty open framework, so you can always fork these and add your own customizations or focuses to these workflows. Okay. All right. One of the other interesting things we've added recently is support for tertiary analysis partners. So we've partnered with a few different companies. You can find them on our at that link at the bottom of the slide. But essentially, what we're trying to do is make it easy for you to go from the outputs from the human variation workflow to basically from your secondary analysis on the human variation workflow, which is going to give you VCF files, amongst other files, to actual more actionable results. Essentially, a VCF file for a human sample might give you millions of different variants, and sometimes it's difficult to determine which ones you should focus on in your studies, especially for clinical patient samples. You might want to have more of a short list of genes, and tertiary analysis partners can help you with that process of filtering and processing those VCF files. Okay. One of the things that I always like to point out to new users is on our epitome site. So this is the epitome.nanoporetech.com site. We do have data sets that are available so that you can download if you wanted to. These include a number of different human genomic samples, but also some metagenomic samples and transcriptomic samples that are available. So for like human genetics, we have genome in a bottle samples, we have tumor normal pair samples, we have data from our newly released hereditary cancer panel, data sets. So, these are all freely available. They're all hosted on Amazon AWS S3 buckets, free to download, free to look through. Some of the workflows, some of these samples, I should say most of them were base called with both high accuracy and super accuracy models. So you can download either the raw data in POD5 format, the BAM or FASTQ files that are already base called, and for some of the for some of these data sets, we've run the samples through our workflows. So, for example, in that screenshot, you can see genome in a bottle samples at the top, and we've run that through our base calling and human variation workflows. And we include the outputs from those workflows as well. So if you want to kind of just get a quick peek at what, a full sample would look like when it's analyzed through one of our workflows, this is where you can go to find some of these examples. Okay. Yeah, so our base calling workflow, as I mentioned before, it's essentially a Nextflow wrapper around our Dorado software. Generally, I recommend just using the standalone Dorado software directly on your system rather than using this workflow. It tends to make it easier, and this workflow is always a little behind on the version of the Dorado software. So again, standalone is going to be always the more up-to-date option. This one has the added complication of, making your Nextflow making your NVIDIA GPU available through Docker or Aptainer, which, sometimes can be more straightforward, but other times can be a little bit more difficult depending on your expertise with your OS and the software. Okay. The alignment workflow is a wrapper around Minimap 2. So most of the features here are features you can get from just running Minimap 2 from the command line if you wanted to. Idea is basically you're just feeding it the output from Minnow, either those FASTQ or BAM files. It's going to basically get the data ready. It's going to give it to Minimap to align, and at the end, you'll get a BAM file with your aligned data set. But you'll also get a report, so that's an extra benefit of using this workflow. You'll get a nice HTML report that will give you sort of an overview of your sample, ideas about your fragment sizes, the distribution of those, quality distribution, quality score distribution, as well as alignment statistics. So it does provide you with extra information that you might have to get by kind of further analyzing your alignment data with a third-party program. So yeah, there are some benefits to using it. So human variation. This is our workflow for capturing the totality of variation in your human-derived samples. Again, the idea here is we're taking our output from Minnow directly into the workflow, so we're not really looking at any sort of pre-processing. And the workflow will detect single nucleotide variants. So, single nucleotide variants and small indels. It'll detect structural variants, STR expansions in your sample, copy number variation. And it can also output a bed methyl file containing your methylation information for the sample. And it can phase your sample using the variant data that it determined. So if you do run your sample through this workflow, you should, if you have these options turned on, get an aligned BAM file that's phased. So you can look at the two haplotypes in your sample, and it will include VCF files for the single nucleotide variants, structural variants. The STR report is quite useful if you're interested in those regions, and copy number variations as well can be very useful. Okay. The somatic variation pipeline is very similar to the human variation. The main difference here is it's going to be looking to take in two samples, a tumor and a normal. It does work with tumor-only samples as well. This workflow uses similar but distinct tools for calling variants. So instead of using CLAIRE3 for single nucleotide variant and small indels, it's going to use CLAIRE-S. If it's a tumor-only sample, it'll use the CLAIRE-S-TO for tumor-only. And it will use Severus for structural variant detection. And Severus also has a panel of normals mode. So if you do have a tumor-only sample, you can use that panel of normals as a stand-in and help you with calling your structural variants in your somatic sample. Okay. So our GitHub page, we have two main GitHub pages. One is our Epitome Labs GitHub page, and that mainly houses our workflows that we just discussed. But we also have a Nanopore Tech GitHub page that has a lot of our other projects on it. For these, I'm going to highlight our Dorado standalone base calling server software and our ModKit software. These might come in handy, so we'll go into a little bit more detail on these in just a second. Okay, so Dorado is the software that we use for base calling. It is integrated into Minnow as well as that WF base calling workflow. But you can also find it on GitHub. And the GitHub is basically the bleeding edge of the release for the Dorado software. So new features, new base calling models, they all make their way there first. And then as the software gets developed, as bug fixes are implemented, once a release is stable, we will integrate it into Minnow, and we'll do some modifications of it. And we will also include that in the Epitome WF base calling workflow as updates make their way into that workflow as well. But the latest features are always on the GitHub standalone version. Okay, what this means in practice is for most basic cases, you really don't need to be using the standalone Dorado base calling software. But as you can see, we do have some subset of features that are only available in the standalone Dorado version. This usually includes newer modified base calling models or the latest DNA or RNA canonical base calling models. They will always be there first. Right now, I believe we're just waiting on, I think we have version 5.2, the latest DNA and RNA models are now in Minnow. I believe the only thing we are missing is some of the 2O methylation models for the different bases. And I think that might be it right now. I think that's the only thing that's currently missing. The other thing that the Dorado standalone software has is it has some additional features like read correction, assembly polishing, and variant calling that we've added to the Dorado software fairly recently. These aren't accessible in the Minnow software. So if you do want these, you'd have to run them through standalone Dorado. Yeah. Okay. And then one of the other standout features, or one of the main standout features for Nanopore sequencing is that you can get these modified base information from your sample without doing any other sort of wet bench approaches like bisulfite sequencing approaches. These are all just there in your raw data, whether you base call them or not. So you can always go back and, base call your sample, your raw data again, and then select a modified base model if you want to. The ModKit is essentially sort of a toolbox of a variety of different tools for working with the BAM files that are produced when you do perform modified base calling with our software. Inside those BAM files, there are MM and ML tags that encode the information about the particular bases that have a modified base and what the probability that that is a correct answer. Those are encoded in the MM and ML tags. But the ModKit can do a number of features. The first feature that it had was to essentially output a bed methyl file. So that basically gives you counts of modified and unmodified bases at different positions in your genome. And that can be useful for some third-party software where they're looking for data in specific formats. You can always convert it. Bedmethyl is a standard file format. It's essentially like a bed style format with like tab delimited columns. And you can always convert that as needed to other formats if you wanted to or needed to, based on what kind of tools and software you want to use. But the pipeline also includes analysis for hemimethylation or chromatin stenciling workflow. It also includes what we consider to be the best practices for filtering these modified bases based on our analysis of the data. And it does include differential methylation calling. So if you do have multiple samples and you want to see how the methylation patterns are different between them, you can always run that data through this software. And before we move on, I just wanted to say that it is, we do mention methylation sort of nonstop in ModKit because it was initially designed for looking at 5MC, 5HMC modifications. But the software, the analyses also work with all of our other modified base calling models, whether you're looking at RNA mods like inosine or pseudouridine. Yeah, it would all work for those as well. Okay. If you go to our resource center, there's a link there at the bottom of the slide. You can find information about community developed tools, workflows, pipelines, research papers. These are all on our website and can be really useful when you basically want to see what other people have done in your field using our devices for different applications. So it's always a great place to stop by and find some of the latest research. Okay. All right. So again, one of the great benefits of our platform is that we can detect modified base information in your samples. So whether you're looking at methylation on cytosine or adenine, or like I mentioned, all of these other modifications like inosine, pseudouridine. If you're looking for these non-canonical bases or modified bases, you can use our software to find them and detect them. We do have, because we're sort of kind of having fun with the whole CPG island, shore, shelf, and open sea idea of human DNA methylation. But we do have a pipeline specifically. So if you really are interested in getting at the totality of methylation in a sample, we do have a reduced representation methylation sequencing workflow and pipeline for looking at data using adaptive sampling to essentially focus in on all of these CPG islands, shores, and shelves in the human genome to really get you a full data set without having to use a PromethION flow cell or maybe multiple PromethION flow cells to get adequate depth of coverage for your analysis. Yeah, I think we can go from there. Yeah, and yeah, as you may be aware, DNA methylation, things like the 5-methylcytosine, is often used in the genome for modulating gene expression. So either silencing gene expression by adding methylation around promoter sequence, or in the case of 5-HMC in specific tissues like brains or stem cells, you can find it plays a role in activating gene expression in those specific tissues. And that's usually where you'll find it most highly expressed as well. It can also help with genome stability, cell differentiation through modulating gene expression activity throughout development. And you can also see it in things like X chromosome inactivation. So in XX cells in the body, you have a silenced X chromosome, and you can find methylation information on that cell again on that chromosome using our approach. And of course, this can also play important roles in uncovering sort of the basis of a disease, especially where maybe there's not a clear structural variant present. You might find a methylation pattern that hints at why a certain condition arose, even when you might have a healthy copy of a gene on one of the chromosomes. They might be silenced. Oh, do we want to go back, Shani, or do we want to?
I think we can just focus on this. Yeah.
Yeah. So when you analyze your data, it's not really just on a per read basis, but you will be looking at essentially a pileup of your data. Other platforms, that's really the only way to analyze your data. But what's really nice about the Nanopore platform is that you can look at all of these methylation patterns in the context of the reads that they're on. So again, we're not looking at shorter fragments. We're looking at, could be 5, 10, 20, 50, 100 KB long fragments. And you can look at the methylation pattern, and this can help with phasing your data. So you might see interesting methylation patterns, but maybe it's only in one of your haplotypes and not the other. But while for other platforms, maybe you're looking at sort of the aggregate methylation pattern for the entirety of your sample, here you can break it down into the different haplotypes and really see on a read by read basis what's occurring. And this can be very useful, not just for methylation in DNA, but for RNA, where you can look at individual RNA transcripts and look at the patterns of non-canonical bases on those molecules. Okay. So ModKit, again, it's going to take in what we've called a mod BAM file in the past, but it's basically just a BAM file with MM and ML tags with that modified base calling information. Again, I think Shoni's probably showed you that in our Minnow software, it's really just toggling a button in Minnow, and now modified base calling is turned on. So this is going to be helping you analyze that data. Again, it will output that bed methyl format file, which is a bed formatted file. And that's going to basically be sort of an aggregation of all of your methylation information in a coordinate by coordinate position throughout your genome. It is integrated into the human variation workflow as well as the somatic variation and trio workflows. So anywhere where the workflow is working with methylation data, it is included. But again, it's also a standalone tool that you can download from our GitHub page, and you can use it from the command line for analyzing or including in your own workflows if you wanted to. Okay, so we're going to go in a little bit more detail with the human variation workflow, since that's probably one of the ones you're maybe more interested in. So if you're looking at methylation information for the human variation pipeline, essentially the pipeline has a number of sub-workflows, and you can select them on the workflow options page. This includes, turning off or on different features like single nucleotide variants, copy number variants, STRs, but also the mod pipeline. For human variation, since it's only a single sample, there's no comparison that can be done. So essentially the mod portion of this is using ModKit to export that bed methyl file for you, which could make it easier to analyze your data with. The alternative, of course, is the BAM file, which is going to be aligned to your reference. So you could, if you wanted to, use that information. And some of the tools that we'll talk about can use that BAM file directly if you want to use them. Okay. For ModKit, there are additional arguments that you can set. And like some of the other tools in this workflow, like Sniffles and Claire, you can specify additional arguments that you want to pass along to it. So maybe if you wanted to, ignore the 5-HMC modification in your output, you can always include that here in the workflow. Similarly, if you do have a more powerful system or an HPC and there's more threads and you think that the ModKit function can be run more efficiently by providing it with more threads, you can modify that option here in the workflow as well. Okay, so some of the main features that are probably most useful are the following. So we have a ModKit summary. So this will give you sort of a global view of methylation in your sample, which may be useful if you just kind of want to, you have multiple sample types and you just want to get an overall view of methylation data in those samples, maybe see global increases or decreases of methylation.
I don't know.
Oh.
I don't know how the slide disappeared. No idea. Sorry, but
No,
probably you can comment on this one.
Oh yeah. Hold on one second. Let me just see.
I don't know. It got deleted suddenly, but I cannot see it anymore.
Yeah, that is weird. Okay, well, I have a copy, but essentially there's a few main functions. One is the ModKit summary. One is the ModKit extract. So summary will give you that global view. Extract can give you information on a read-by-read basis. So again, that might be more useful if you're looking at like transcript level data. The ModKit pileup is what we use to produce that bed methyl file format. And it's basically another way of summarizing your data, but it's going to do it across all modified positions in a bed file in a coordinate by coordinate manner. And then we have a ModKit DMR, which is for differential methylation scoring of different regions of the genome. And as I mentioned, we use methylation a lot, but some of these tools can be used with other modified bases or non-canonical bases in your data set. And then we do have a way of converting the bed methyl file to a bigwig file, which can sometimes make it easier to visualize in IGV. Okay, now moving on to this slide. All right. Yeah, so here's just a quick list of sort of troubleshooting and common issues that you might run into in ModKit, and these might be more useful to review as you run into them. But there are certain parameters and things like running at the bottom the --CPG. So if you wanted specific types of modified bases in your outputs, you can specify them. There's multiple ways of filtering and modifying this data set as it's being analyzed. Basically sort of like how you want to set thresholds for, calling a site modified or unmodified. By default, ModKit will handle that analysis itself and will apply it to the runs that it's doing. But if you wanted to, you could manually set those and derive those yourself, depending on basically how stringent you want to be in calling sites methylated or unmethylated or modified, depending on the model that you're using. Yeah, I think it's good to revisit when you start using the software. Okay. Yeah, so there's a number of ways of looking at the modified base information in your files. Sort of the easiest one, and the one that I usually go with, is something like IGV or JBrowse. You can drop your modified, your BAM file with the modified base information in it. Once it's aligned to a reference, you can just open that up in IGV like you would open up any other BAM file. That's not aligned to a reference, and you can look at your methylation patterns just right there in your sample. You can also use the bed methyl or that bigwig formatted version of the bed methyl file if you wanted to look at it in sort of a more global view, so you can zoom out a little bit further and still see the details. There's also tools like Methyl Artist, and I believe we have a slide for that coming up, so I'm not going to jump too much into that. But yeah, there's a few different ways that you can visualize and even in some of these tools do some analysis of your data sets. Okay, yeah, we can go on. Okay, yeah, so as I mentioned, IGV is one of the ways that I will usually turn to once you have an aligned file. It does work with BAM files. It works with the VCF files. So you can kind of load up all of your data sets into IGV and kind of look at them all together. Basically see, where you have structural variants or single nucleotide variants being called, and where you have methylation patterns. You can kind of look at it all together all at once. And it's as simple as just going into IGV and going to the color alignments by, and then choosing the particular base modification that you want, whether you're looking at the 5MC, 5MC, 5HMC, 6MA, or really any of those modification calls. Okay. Yes, so you can, as I mentioned before, there's ways of converting bed-methyl file to other formats like BigWig and your bed graph. So you can do these conversions. So if you wanted to, make them different types of tracks that you can open up along with your BAM files, you can do that using that little snippet of code for the command line. If you're familiar, use it with the awk tool. You can use that to just do a quick conversion of the bed-methyl file, and then it'll basically allow you to treat it as a bed graph in IGV. Methyl Artist is a more fully featured software tool for looking at and analyzing methylation data patterns. It does support mod BAM, or essentially BAM files with those MM and ML tags. So it does support that directly. So you don't need to produce bed-methyl files for your data, or do any sort of other conversions or extractions of that data from that file. You can essentially provide Methyl Artist with your aligned data directly. Methyl Artist is great at making these really nice, informative, and easy to understand charts. So here you can see it's doing a comparison. It looks like between 4 different haplotypes, showing ones that are hyper and hypomethylated for these regions. And you can see that on the top part of the chart, where you're looking at the reads as aligned to the reference, showing you the coordinates, as well as the charts below showing you sort of the percent modified at these different positions in different ways, either in sort of a smaller binning or larger binning. And it does include functionality where you can do some comparisons of methylation between different sites as well.
Okay, I think that's what we had about, methylation specific data analysis part. I already, discussed few of the papers yesterday. Want to go quickly over some community web page and help, which we have, and I think these are very useful. And after that, I think we can take a look at the data, which got generated, and we can see, if we help you to start a run, either Epitome, if it is installed yet, or, any help you need regarding installation, we can try to do that after this. So if you go to Nanopore community website, you can create ID to log into this community. And I think the Nanopore community website, what we have, is quite useful. It's kind of for all the learning purpose. We do have lots of documentation specific for different types of applications or different types of library chemistry. I have already shared a few relevant links. I will try to gather some more and will share. This is quite an important learning tool, I would say. Yeah, here, like, it is an example how we can use it. There is a tab called Getting Started. Like, if you can click there, you will get the idea, like, what this tool or this web page contains, what are the resources we have here. There are like protocol tabs, software downloads, product updates. These are quite useful. Specifically, I would say the protocol software downloads and whatever the updates we do, and you can have all the ideas, all the latest information from here. There is also Nanopore Learning. So, on the day one, I was showing a YouTube video, like where we saw like how to do the loading. So we have tons of, learning videos as well. I'll also, once again, gather some, useful learning videos and we'll share. So once again, this is quite helpful. I think this is something what we already watched on day one. There are lots of courses. Those are also very useful. If you are interested, you can also review that. And the documentation section, specifically if you type here CFDNA, you will get all the relevant information which are publicly available from Nanopore related to cfDNA sequencing. So that's quite an useful tool. And also there is community support, questions are posted here. And there is, FAQ section. Quite relevant information can be obtained here. If like any trouble, any issues happen, let's say during run, during analysis, even like related to warranty, feel free to email me, feel free to email Matt if it is bioinformatics related, but also you can contact support. So based on the, what the question is, we categorize like who is gonna take care of that. For like major troubleshooting, problem solving, even, project discussion, it would be either me or Matt, but sometimes, there are other technical support team who can be also reached out if you contact support. So this is quite important as well. Yeah, I think that's what. Okay, let me stop here. Yeah, so that's what we have in terms of data analysis discussion. And I think right now what we can do, Teddy, and Shweta, and like anyone who wants to, if you have any kind of particular question related to data analysis, starting from Mino or like epitome or any specific workflow, please feel free to ask. We'll try to help and answer here. And after that, we can see like how exactly like with the present run, how we can help to start the analysis. I think that's what we can do today. Yeah.
Great. Thank you. I guess the first question that I have is with respect to the reference files, and if Oxford Nanopore has any curated lists, especially like for the epitome workflows where they're more all boxed up.
Yeah. So for some of them we do, because it kind of makes sense based on the workflow. But for ones like human variation, we do have recommendations for a reference that you could use, but we don't have a preferred reference. For other workflows like the single cell workflow for 10x, we do have a link for a specific 10x human reference. I think 10x also has a mouse reference that you can download. So basically it's like a prepackaged and compiled reference and annotation in a specific format that 10x, I guess, likes to use. So we use the same files. You can find the link for the reference for the human variation. It should be near the bottom of the README. It's also, where is it? It is, yeah, we have a section on the human variation. I can show you. Give me 1 second. Open it up so we can all kind of take a look at it. Let me share my screen. That's it. Okay, can everyone see my screen? Okay, yeah, so for human variation, we do have a recommendation down here. So if you wanted to do, was it HD19, I think, or like the version 37, or if you're HD38, you can use the this one as well. So we have 2 different recommendations depending on what sort of reference standard you want to adhere to for human samples. And then we have a link to a really nice write-up by Heng Li about why use certain references over other references. And it kind of goes over some of these sort of features and issues that might come up, like including alternative contigs. So having like basically multiple copies of specific regions of the genome that are highly variable, and including those, which can kind of do interesting things when you're trying to align data to it, right? Because you have reads that might align to 2 different regions or equally well. So it's good to kind of think about those ideas here. And so he has basically 7 points that he thinks you should always think about before you use a reference. For human references, we sort of have a lot of choice there, right? So we have like HD19 and 38 if you want a more standards or sort of older standard reference, but there's also the new like T to T references, like this CH13 or CHM13, which is one of the T to T references. So yeah, you do have choices here. The other thing I wanted to mention, which is kind of related, is, not here. The human variation workflow, it can really work with any data set. You just have to provide it with a reference. But some of the tools, like the copy number variation or STR expansion analysis or annotation, are assuming it's going to be HD38 or HD19 references, or for STR, it has to be HD38. So if you use other versions of these, you have to basically make sure you turn those features off. So if you're using like a T to T reference, just want to make sure that you turn annotation to false and that you are only looking at these particular categories. Copy number variation won't work with our workflow as it's written. Same for other organisms. And for other organisms, you just want to make sure that you do include all contigs because the workflow has some hard-coded chromosome labeling. So it's going to look for exactly the number of chromosomes that we have. So if you're using mouse or another organism, they're not going to be the right naming or number. So just something to think about. Does that answer?
That's great. Thank you.
Okay.
And then I guess just to continue with my next question. Mm-hmm. So with respect to the methylation analyses, specifically in our context, looking at, the methylation profiles of cell-free DNA, I think one of our longer-term goals is to do some kind of, methylation-based cell type or tissue origin classification. And I was wondering if you have any, if you know of anyone else doing that, or if you have any guiding points as far as where to get started on that. I know it's kind of downstream of all the, actual methylation calling and alignment and all that kind of stuff.
Yeah, there's, I believe I reviewed some papers with you, I think, right? Maybe for cell-free already, but we could always send you some additional links. But There's definitely been a few papers that have come out that have been looking at cell-free DNA and methylation content and using it for sort of essentially monitoring things like tumor progression in patients. Maybe you've seen the Billy Lau paper, which kind of has like a great review of this, basically showing sort of this cancer-associated methylation profiles over time, showing it as like the patient's getting chemotherapy, surgery, and then as they see a resurgence of the cancer, and you can see the levels kind of dropping and then increasing for that particular patient. There are a few tools and approaches that are out there. They're not, I don't want to say that there's not a lot of like fully featured pipelines, essentially, for this kind of thing. There is one from the De Ridder lab over in the Netherlands. They have a nano RCS pipeline. This pipeline is for cell-free tumor DNA profiling, so that might give you some insights. Their particular pipeline, one of the key sort of features of their analysis is they're using rolling circle amplification of the cell-free DNA. So methylation information was one of the things that you would lose by amplifying it. But they're doing the amplification specifically for the purpose of getting really low frequency variance from that circulating data, from that cell-free DNA. So yeah, there's sort of different approaches and ways of kind of coming at this. So, I would probably just focus in on sort of Billy Lau's group for methylation profiling, and maybe the De Ridder group's nano RCS pipeline, if you're interested in potentially looking at sort of very low frequency. I think they said they got it below 1% frequency. I think it said 0.24% was the lowest they were able to confidently call. And I can put a link for that in the chat if you want. So you just take a quick look at that, or, so you can take a quick look if you want. I don't know. I don't think, I have to double check. I don't know if Billy Lau has like a fully sort of put together pipeline for his analysis. He does describe how he's doing it in the paper, but I don't think there's an archive. And let me send you the paper that goes along with that, the link for that paper, for that nano RCS pipeline. I have to take a look to see. I double check the Billy Lau paper. He had a recent one that came out, or at least it's on bioRxiv for this year, earlier in the summer, looking at methylation. This was from cerebrospinal fluid, so not blood, but it's a similar approach, right? Looking at the cell-free DNA, and I think they're looking at brain metastases from like a lung cancer. So again, it could be useful and could be helpful. Yeah, I think so. Yeah,
that paper we shared earlier.
Oh, you already shared? Okay. Perfect.
Yeah, I think that is the same paper, right, Teddy? Like we are talking about the 2025 bioRxiv. I think it's the same one.
Yeah. I'll give these all a read and go through. I think the main difference with our particular approach, while it is CSF as well, which is helpful, we seem to be doing kind of, aggregate methylation percentage between cancer and or tumor and healthy. Whereas really our goal is to say, based on, or from what I've read, based on methylation profiles, you may be able to identify, whether this is of neuronal origin or, leukocyte origin for the cell-free DNA. That doesn't necessarily, but they're both from a quote unquote healthy individual from a cancer standpoint.
Yeah. It's not quite what you're doing. But there are, maybe you've seen like the Robin paper from Matt Luce's group. I can put a copy of that in the chat again. This is, they're essentially doing, I guess one way to think of it is they're doing sort of like intraoperative sequencing of patients. Actually, I think, is this the Robin paper? I think that's Robin. For brain tumors. So basically they're using sort of like basically using profiles that already exist for cancers in the literature to define, specific targets of interest to help them categorize based on it. And it's Robin, and now I'm trying to think of the other one. There's one more group, Marlin.
That's the Dana-Farber?
Is it Dana-Farber? Let's see if I can find it.
I think Dana-Farber, yeah.
Yeah, do you have a link for that?
Yeah, I can.
So there's a tool, Marlin, which I've spoke with another group I'm working with because they're interested in leukemia. And let me see, did she put it? I have it right here. So it's a Nature paper from 2025. Essentially, they're using, again, methylation information to categorize the type of leukemia that the patients have. So basically to help with treatment options, right? So again, you can, these might not be exactly what you're interested in, but they might be really great places to get ideas. So if you want to develop sort of your own system for your own, sort of diseases or cancers of interest, you can kind of see what they've done. And I would say that I think both Matt Luce and the group at Dana-Farber, I believe, are both pretty open. I know Matt Luce is definitely open to, like, if you have questions, you can always reach out to him. But yeah, those groups, and I don't think the other tool. Yeah. So these might be sort of useful approaches and ideas. There's also another short. I can't think of the other one. There's like Robin, and there's one more pipeline for looking at sort of intraoperative tumor analysis, which again could be another useful. Let me see if I can find that. But we can.
Thank you. And then I guess as a brief question while you're looking. So we, in our initial real-time base calling for the run that's currently going on, we did not click the methylation option. So our plan is to now just do kind of the post hoc methylation calling with Dorado and the pod5 files. The question is as we're setting that up, and this may have been mentioned, I just forget, is there a consideration of choosing which model we should use impact the methylation calling, like the hack, soup, and which, or basically which one should we do now that we have the option to just run it afterwards?
Yeah, it's we do have. Let me see if I can bring it up. We have an accuracy page. I can show you. Don't know. I'm not sure if it includes it, but basically, we have, we have charts here showing you the difference between high accuracy and super accuracy models. And it's hard to read, unfortunately. But I think what the chart is trying to convey is that between high accuracy and super accuracy, you're getting to the point where you're not seeing sort of robust jumps in accuracy anymore, especially for single nucleotide variants at really all levels of depth. You're not really seeing huge jumps as you go from like lower to higher depth or from hack to soup. if you have a 30x genome, it's 99.7 to 99.8 F1 score change. So, you're not really going to see big jumps there. But what I did want to highlight, well, I guess before we move on from there, where it does make a bit of a difference is if you're looking at some of these indels. So if you're interested in some of the smaller insertion and deletions, the accuracy improvement with higher depth and the soup model, may be enough to convince you to go with that model. So I think I wouldn't think of it just in terms of the modified base analysis. But when we do perform modified base analysis in QC, we tend to use the super accurate model for both RNA and DNA. And that's where we're getting the scores that we report. So if you do high accuracy, your scores are going to be probably just slightly lower than these. So maybe instead of 99.5, maybe it'll be 99.3 for like CPG methylation. But yeah, so that might influence your decision a little bit. That we tend to use super accurate for these. The other thing to consider is the sort of the volume of data and samples that you're working with. For most of the groups that we work with that are working on really large scale projects, they tend to use high accuracy almost always. So they're going to use high accuracy calling because for all of the variants that they're calling, it's good enough. It's just about the same as soup, but it's a lot less time is spent on the base calling. If you wanted to, you could always take one of your data sets and, you could base call it with both approaches and just kind of compare the results, see, how much disagreement there actually is. But we do have data sets, like I said, on our Epitome Lab site. So you could download already base called data sets for high accuracy and super accuracy for genome in a bottle samples and compare those results. So you can, you don't have to do it yourself. You could look at our data that we already provided if you want to kind of get a less biased take on it. But yeah, that's what we do internally is we do super accurate, but high accuracy works almost as well.
Do you have any other question related to the data analysis part? Otherwise, we can try to figure out like how you can start a post-run base calling if that is already set up, like the Dorados standalone, or like any help regarding installation, if you would like to have or need.
Yeah, I can share my screen with the mid-run report just to kind of go through the sizes and stuff. As far as the actual setup goes, I think we can just wait until the run is completely finished. As long as, it sounds like I've downloaded the next flow and been able to access the human variation workflow fine. And then Dorado seems like a standalone. So
yeah,
I think that, so long as that's the case,
if anything comes up that time, reach out.
Let me share my screen on this one. All right, so I think the one thing we were mentioning yesterday was with respect to kind of the size of reads generated, noting that it was pretty low, which is understandable given the amount of sample we loaded, as I understand it. I guess my first knee-jerk reaction is that then justifies running the super accuracy on everything because of how small it is. I don't know if there's further consideration, but given that we're not working with that large of data for this particular run. Seems like why not? And then running through, again, this is now our updated histogram.
Yeah.
Continues to look representative.
Yeah, it's like very similar to what we were observing yesterday. Does not look like anything got shifted that much, right?
I think so.
Yeah.
If it influences your decision in one of the papers from Billy Lau and his group, they are using the SOUP models, so super accuracy, for their base calling. Yeah.
So Matt, like Daddy's lab, like Tim's lab, they have P2 solo. And, you know, the thing is like, you know, we don't have any comparison of like a keep up document specifically for the P2 solo, because the computational spec is so much variable across different labs. So that's
Do we know
the reason.
What GPU they have?
Do you have that information handy?
It's like the NVIDIA RTX 5090. Basically, all the specs on the Nanopore website, we match them to a T.
Yeah, no, that's a great one to use. And this is in a desktop, right? The desktop version of the GPU? Like for workstations?
I mean, it's in a laptop. I don't know. Let me see if I can.
Oh, okay. All right.
Pull up
I just want, because it has, because the laptop is going to have a little bit less RAM and it's going to have less cores than the workstation version of it. Okay. Yeah, so if you're using the 5090, I don't know if the laptop one, we have here's let me see if I can find it.
Let me see if I can.
Yes, there's some community developed like GPU benchmarks. I don't know if the laptop 5090 is in there. But I think the 5090, the laptop version is more like a 5080, I think, for the workstation, like a desktop equivalent. I think it has like half the cores and it's got less RAM than the 5090 in the workstation. The workstation one is more than capable of keeping up with soup-based calling for two positions, whereas the like a 5080 or 5070 doesn't quite, but nearly keeps up with a single flow cell at soup, and that's about 150. I think it can keep up with about probably somewhere between 30 and 50 gigabase pairs per day. So just something to consider. But it's, it's worth taking a look at. I don't know how much, if you go back up to the top for the total amount of data produced so far, 38 gigabases. Yeah, I mean, if your library is not, if it's cell-free DNA and it's not producing as much read data as like a whole genome sequencing, then it might be fine for keeping up with that for soup. If the software falls behind, it usually just starts packaging the pod 5s as being skipped. So if you start seeing skipped folders, you're kind of overtaxing your system.
I see. And I guess as a question of the pod 5 and FastQ packaging, we've been doing it by the hour, so we have just a bunch of stacked files.
Yeah,
I noticed that the Epitome has like a tool for concatenating all of the fast Q files, is that something that needs to be done before running it in any of the other workflows, or
No,
can I just feed it in the folder directory?
You can feed it in the folder. It's the FastCat, I think, is the program that we have. It does concatenation, but it also produces some statistics that we use in some of the workflows that we stream. You can just feed it like all of the different, like if, we have some groups that do like every 10 minutes or every minute or something. So there's like thousands of FASTQ files and BAM files. And, I think concatenating BAM files particularly is kind of a pain. But FASTQ is pretty simple and straightforward for concatenation if that's what you're using. You can do it from the command line. You can use FastCat, or you can just use the workflow's ability to concatenate things instead. So you can give it a folder full of BAM files or FASTQ files.
And that's the same with the Dorado. I can just feed it the folder of OD5 files.
Yeah, exactly. You don't have to list each one, and you can just pass it the folder. Yeah, and it'll look inside that. I think most of them can do recursive searches, which you may or may not want. So just be aware.
All right. I think the only other thing to highlight here, the quality score, the number of failed bases, the peak has increased since yesterday. I think the question is how or what to consider when entering a min Q score. I noticed we were going through an example report yesterday, and I noticed that the min Q score was 10. Now it's set to 9, and just, I guess, what we should be considering in that regard.
Yeah, I think for this run, I did not change it is the default one, 9. That's how I set it up. You can do you can change it to 10, but I think for CFDNA, I feel like this is fine. This is what that's what I would do. Do you have any suggestion, Matt? I think most of the, because probably you have worked with more CFDNA labs. Any other suggestion you have regarding that?
Yeah, I feel like I would keep it. I'd keep it with the default settings for sort of this first pass QC. There's no reason for you to use anything below the 9 or the 10, depending on the base calling model you're using. You can safely discard that. Data. Let me think if there's anything else. Some groups go lower, even than 9 or 10 for the soup base calling model. It might be worthwhile for you to kind of assess even the failed reads and just kind of see what the quality looks like. There might be very good reasons why they're failing. Maybe the quality is just very, very low overall, or there's nothing sort of redeemable about the sample. But there could also be a good reason to kind of go maybe to Q8 or Q7 data. I think when Billy Lau does his analyses, I think they go down to Q7, I think, for quality score filtering. But there's other groups that will basically filter by alignment. So if something doesn't align to the reference or align unambiguously to the reference, they'll discard it then. So there's a few different ways that you could do filtering here, especially for this selfie DNA where they are shorter reads in general. It could be helpful to, sort of assess the quality maybe with a secondary step instead of just a Q-score threshold. But I think this is a great place to start. But yeah, I would definitely take a look at some of the failed reads and just kind of assess them, see if there's anything that you can recover from them. And if not, just, feel free to discard them. Because you are getting a lot of data that's, well above that. And if you switch to the super accurate model, you might see you're getting more like a Q20 mode or higher for your data set. So, yeah.
All right. I think that's all the questions I had And I feel, as far as the setup of the actual software, I don't have any additional questions. Anyone else?
Okay, so I think what we can do, once the run is over, like you can share the run report with me. And if you have any additional question, please feel free to ask. And then we can start working on the analysis. If, there is any issues during the analysis also, please reach out. And then we can again meet probably at the beginning of December, just to go over this data or how the analysis looks like. Yeah, that's what we can do for this pilot. And I'm gonna share an update of the training, like additional any kind of relevant links, any paper suggestions we have from our side. And I think probably one form needs to be sent to you and one survey link. I'll do either at the end of today or on Monday.
Great. Thank you.
Yeah. Thank you so much. Thank you. And I'll be in touch soon. Thank you. Have a good
Thanks for all your help.
Bye.
I think we all really enjoyed the trainings, and we're really looking forward
Thank
to.
you, team. Yeah, actually, it was great interacting with the entire team. The work you do is excellent and very inspiring.
Thank you for saying so. We're excited to explore new horizons, and with Oxford Nanopore's help, I think we have some novel angles here that could potentially help people, which is our ultimate goal. So, thanks everyone for helping set it up. Teddy, also thank you for all this, and we look forward to being very good, better customers in the future. So.
I'm sure. And also, Teddy, I sent the RNA sequencing protocol. And if you think you want to explore it in the future, let me know. We can talk more about it. And I think Sam can help you with the purchase of the flow. You have the flow cell, like the kit, if you are interested in future.